Welcome to this first lecture on cell cycle. And we are having this lecture on Let's Crack NEET UG English. Before we start the cell cycle, which is our topic, and this will be completed in couple of sessions. This is just going to be the first session. So before we actually come to the topic, let us first talk about the platform which we are using for this lecture. It is Unacademy Let's Crack NEET UG English. And about myself, I'm Dr. Neela Bakore. I'm going to be teaching you biology. I have been teaching biology for almost 23 years now, and botany and zoology both I take when we talk of biology. I have taught in India as well as in the US, and I have been mentoring students who are preparing for pre-medical entrance exams like NEET. Unacademy platform is the largest learning platform which we are using. And to use this platform, you need to do a very simple thing, and that is you have to install an app. It is called the Unacademy Learning app. You just install this app, and as soon as you install this app, you will get benefited. Now, what exactly are the things which you will be getting through this app? There will be daily live classes. The top educators are going to take live class. I'm sure you have experienced the difference between a class which is like a recorded session and a class which is a live class. So in live class, you get to interact with your tutor. You are able to get the doubts solved. So those classes are conducted live on a daily basis. The classes are structured in such a manner that your complete syllabus for NEET, whether you're in 11th class or 12th class or whether you're a dropper, all those chapters will be completed in a very structured manner. Once you study from the top educators and you want to assess yourself whether you've understood everything or not, then the best method is to take a test. So there will be live tests, there would be quizzes which these educators will be conducting for you. So it will help you assess yourself. Sometimes we find out that one particular thing is not clear and you're committing mistakes. Then it gives you an idea that you go back to that particular thing, revise that concept and take the tests again. You will get unlimited access to all the educators, the free live lectures which are there, which will be available for all of you all the time. Live classes are conducted by a large number of educators, those who have experience in training and mentoring students who are preparing for NEET. Let us talk about the financial part a little bit, the fee structure. The fee structure is flexible. You get variable options depending upon what exactly are you looking at. Suppose you just want to give it a try and want to see whether this is really working for you or not. There is just a monthly plan and the amount is just 5,400. But if you're planning for 1 year regular study, then the best option is that you go for a 12-month plan. Suppose you're in 12th class, so you want the complete 12th class syllabus to be done before your exams. So it is better to go for a longer duration plan. In longer duration plan, there are two main benefits. One is financial, which you can see here. That is, the amount is just 27,000 and which is after a big discount. The second is, on a long-term platform, you are able to get or remain associated with the educator on a daily basis for a long period of time. So if you learn from a teacher for a long period of time continuously, you get associated with that teacher, you understand the way of teaching, and then it becomes very easy instead of switching from one place to another. If you are, say, in class 11, then you have to plan for your 2 years before you appear for NEET. So you can even go for a 2-year plan. And again, there will be financial benefit. and you will again learn from the same educators. So once you are used to a particular method of teaching, the same educators will be with you. And if you take these subscriptions using my code, then you will be able to avail 10% discount on that. So don't forget to use this code whenever you subscribe for whichever plan. So think about it and select a plan which is more suitable for you. Let us come back to our topic. The topic that we are discussing is cell cycle. Now, what exactly we mean by cell cycle? I have given you a technical definition here. The definition says that it is the sequence of events. And what are those events that we will take up in some time? In which or by which a cell duplicates. That means here we are talking about the division of the cell. Duplicates its genome. What exactly is this genome? Genome is the genetic material which is on one set of chromosome. So this word is important. Genome is all the genes on one set of chromosomes. We will be talking about this in detail in the later part of this lecture. So there is genome duplication, synthesis of constituents. Whatever is required by the cell, that has to be synthesized, and then the cell is going to divide. This is what happens in cell cycle. So this is a technical definition which we were trying to interpret. In very, very simple language, if you have to talk about what exactly is cell cycle, can we not write that cell cycle is the life cycle of a cell? What would a cell do in its life cycle? That is what is cell cycle. So, before we actually come to the details of the cell cycle, we will be talking about some basic things which would be required, like structure of chromosome, what is the cell cycle, what is ploidy, so that when we come to actual cell cycle, it will be easier for us. Okay, this is the question. What is the cell cycle? So cell cycle is the life cycle of a cell. And what happens in this life cycle? A cell is formed. This is the first step. How is the cell formed? The cell is formed, or it arises from the pre-existing cell. This was told to us by Rudolf Virchow. In junior classes, we have studied cell theory, which was given by Schleiden and Schwann. There was a postulate which was added by Virchow that all the cells, they arise from pre-existing cells. So a cell is formed. This is step number 1. What will a cell do when it is formed? When the cells are formed, they are tiny. They are known as daughter cells. So they need to grow to their normal size. And when they grow, they have to accumulate cytoplasmic content. They have to have more and more organelle. That is what is the constituent growth which is taking place here, and then the cell is going to divide. Once the cell divides, daughter cells will be formed. That means from here we go back to the same point, and we are using the word cycle. What is meant by cycle? When we use the word cycle in biology, it means the place from where something begins, it is the same place where it ends. So this is what is cycle. So cell is formed, it grows, it divides. New cells are formed, they grow, they divide. This is how the cycle continues. So now we know what exactly is cell cycle. Let us talk about chromosomes. We use the word genome. What exactly is genome? We talked of genes. What are genes? What is DNA? Where is this DNA located? So we need to understand this chromosome thing, because everything, the genetic material is inside these chromosomes. So that is our first thing which we need to know. Chromosomes, these are rod-like structures And these rod-like structures are formed from thin threads. Just to give you a recap of what you have already studied is a simple cell. We have been drawing simple cells. This is the plasma membrane of the cell. And inside that there is this nucleus. And inside the nucleus we draw thread-like structures. What are these thread-like structures? These are chromatin fibers. Now, what is chromatin fiber and how is chromosome different from it? So, let me draw this thread here. This is the chromatin fiber. Now, in this chromatin fiber, there are going to be some changes. What are the changes? There will be condensation and dehydration. Condensation and dehydration. Now this condensation is not like the condensation of water vapor. Here we are talking about condensation, that means thickening by loss of water, and dehydration is loss of water. In few minutes we will study the detailed structure of chromosomes, so you will know from where is this water lost. So there is loss of water. Because of this, the thin thread-like structure starts to appear like a little compact structure. Further condensation takes place and now the structure appears like a rod-like structure, which is known as the chromosome. Okay, so when we talk of chromosome, where is it coming from? It is there in the nucleus, but only thing is it is in the form of threads, and these threads are nothing but chromatin fibers. When they condense, when they become thick, you start seeing a distinct rod-like structure, which is the chromosome. So now can we see the detailed structure of chromosome? What exactly is inside it? This is what is in the chromosome. I've drawn this diagram for you, I've labeled it for you, and we will discuss few important things. There is a wall around it. See, a basic chromosome when we talk of, we normally just draw the lines or a little dense structure. Very rarely we go inside this. And this particular part is essential for many chapters. One chapter, that is cell cycle, we would need this information. In the chapter cell structure and function, again we need to talk about chromosomes. In 12th class, we have two chapters. One is the principles of inheritance in genetics. We need this structure of chromosome, and then there is DNA replication. So for that we again need chromosome structure. So you study it at one place and then you use it at 4 places. So what we do is, whenever we study chromosome, we do it in detail, so that next chapters when we come to, we would not be going in detail because we already studied it, so that saves us time. So let us come back to this structure. It's that thick rod-like structure. Its wall, the membrane, is known as pellicle, and it is made up of protein. Normally, whenever we talk of membranes, biomembranes, they are proteins and phospholipid membranes. But here it is pellicle, made up of only protein, and because of which it is highly flexible. Because of this protein. So this is one thing. Then inside this chromosome, we find some threads, the chromonemata threads. Now if suppose I enlarge only this much part of chromonemata, what is going to be visible to me is, we would see histone proteins. There are proteins. Do you have an idea how much DNA do we have inside one cell? A cell is microscopic. Inside that cell, the nucleus is even tinier. And these threads, where our DNA is, If you connect all the small pieces of DNA from all the chromosomes, the length is 2.2 meters. 2.2 meter long DNA is fitted inside that tiny nucleus of a tiny cell. So how is it possible? It is possible because our DNA is packaged, it is coiled, it is highly coiled. So where is this DNA? So this histone proteins, around these histone proteins, we will find that our DNA is wrapped. So the DNA is coiled around like this. So if we enlarge this chromonemata thread on only an area, this is what is going to be visible to us. And such a structure is known as nucleosome, that we will take up later on. And this thread which we have drawn is DNA. So where is DNA? When we say the DNA replicates, where exactly is this DNA located? So now we know where this DNA is located. This is one thing. The next important thing is, I'm going to underline this. This is the chromoplasm. Like in the cell, we have cytoplasm. In the chromosome, we have chromoplasm. That same jelly-like material. And remember we talked about condensation and dehydration? So it is from this chromoplasm, when water is lost, then that thread-like chromatin fiber becomes thicker and we start seeing it as chromosome. So now you know from where the water is lost. It is from the chromoplasm. So one is chromonemata. We now know where our DNA is located. We know where this water is lost from. And the third very important thing is this primary constriction. There are two types of chromosomes mainly. One which has only primary constriction, and there are few which have one more constriction, which is called the secondary constriction. We will study these chromosomes, different types of chromosomes later on in the other chapter. But here, because we want to talk about the cell cycle and cell division, our focus is this primary constriction. This primary constriction is also known as centromere. And around those centromere, you find a sheath, this yellow colored sheath. This is called the kinetochore. This is again important. What is the importance of it? Spindle fibers are attached to kinetochore. So this is the place where the spindle fibers will be attached. So when we talk of cell division, we need to know about DNA division. If DNA's division or replication is to be understood, we need to know where it is. If it is in the chromosome, what exactly is the chromosome? So that when we come to the division part, it will be very easy for us. So I have given you some basic information of chromosome which we require for cell cycle and cell division. Some other parts we will take up in the next chapter. Okay, we need to also understand a word which is called ploidy. This is also required for our cell cycle, cell division thing. Number of sets of chromosomes in a cell makes it ploidy. You have heard of haploid, diploid. Now what exactly is this? Let us take our example. In humans, our cells are diploid. What does it mean? According to the definition, it means we have two sets of chromosomes. And how do we get these two sets of chromosomes? We get these two sets of chromosomes from the gametes. Say this is the male gamete, that is sperm. This is the female gamete, that is the egg. So male gamete brings one set, the female gamete brings one set, And when fertilization takes place, the two sets come into the zygote. Okay, so the gametes have only one set. So if an organism has only one set, we will call that organism haploid. Here we are talking only of gametes. Okay, gametes are always haploid. So, but an organism, if it has only one set of chromosomes, then that will be termed as haploid. Some lower organisms like lower alga, phytoplanktons and those. Higher organisms, they have two sets like human beings. So we are diploid where we have two sets of chromosomes. So if there is only one set of chromosome, it is represented by N. One set, only one set, it is written as N. If there are two sets, it will be written as 2N. If there are three sets, it will be written as 3N. So what are we going to call if there is only one set? Haploid. This is what is written here. So N is haploid. What will be 2N? Diploid. What will be 3N? Triploid, and so on. So now we know what is the ploidy, and it is important that how this ploidy is achieved. Suppose we are talking about the zygote, and here the zygote has two sets now, one received from the male gamete, one received from the female gamete. Now this zygote is going to divide to form two daughter cells. How many sets should they have? The most common division which we will be taking up in the later sessions is mitotic division. I am taking an example here of humans only. Our two sets have 46 chromosomes. One set has 23, the other set also brings 23, and that's how we get 46 chromosomes. So now when this zygote divides, one cell should get 46 and the other cell should also get 46. How is it possible? The parent cell has only 46. It is like if I have a piece of paper which has the complete information about, say, one topic, and I have two students, and I want that this complete information should be passed on to both the students. What option do I have? I need to get a photocopy of that, right? So similarly, if this cell has to pass on the complete information to two daughter cells, it has to make a copy of its DNA, right? So this is what is going to happen in cell cycle. So this is why this ploidy is so important. So now we have understood chromosome structure, we have understood what this ploidy means, and now we will use this information for our main topic, that is cell cycle. So this cell cycle is basically completed in two steps. One is interphase and two is M phase. Interphase is long. Interphase is further divided into three sub-phases, and they are written as G1 phase, S phase and G2 phase. G1 is also known as gap 1 phase. G here stands for gap. S stands for synthetic phase. Something is going to be synthesized here. And what is that something? It is going to be the DNA. And G2 is gap 2. So this is interphase. It is very long. M phase, M stands for multiplication. And normally this multiplication is by mitosis, which is the most common type of division. In M phase, again, there are two subphases. One is karyokinesis. Karyon means nucleus and kinesis is division or splitting. So this is division of nucleus. And the second phase is, or second part is cytoplasmic division. So that will be known as cytokinesis. This is division of cytoplasm. Karyokinesis is completed in 4 sub-phases. Because we are looking at cell cycle at a glance, it appears like how many phases are there. But when we actually take up step by step, it automatically becomes clear and it is not at all difficult. The first is prophase. Second is metaphase. Then is anaphase. And the last is telophase. So in these 2 steps, that is interphase and M phase, our cell cycle will be completed. Now let us see the cycle. And what is cycle? The place from where it starts, it is going to end at the same place. This is the one which I have drawn for you. And you can see all the stages which we have written down, or all the phases which we have written down previously. So from here, if you start, it is interphase. So first the cell gets into G1 phase, then it gets into S phase, and then it comes into G2. So this complete part is interphase. See, I told you that this is the longest part. So this is interphase. G1, S and G2. And then there is M phase. In M phase, this much part is karyokinesis. This we have written here, karyokinesis, and then cytoplasmic division, cytokinesis. So now if you start with a cell and say we are starting with a diploid cell, what happens? It gets into G1. What is it going to do in G1? It is going to prepare itself for S phase. That means S is something very important. What happens in S phase? Synthetic phase. In synthetic phase, DNA replicates. DNA is going to divide or replicate. Now, do you know how DNA replicates? Our DNA is a double-stranded structure. The structure was explained by Watson and Crick. So this is one thread of DNA, and then there is one more. This is the other strand. So now when this DNA has to divide, these 2 strands, they separate. We call it opening up of the DNA. And we start seeing a fork. It opens up and one strand appears like this, the other strand appears like this. And on these 2 strands, new DNA will be synthesized. So here will be DNA, we are not going into details of that, but here will be now one DNA, and here will be one more strand, and now these 2 will coil and these 2 will coil. So from one double-stranded DNA, we will get 2 double-stranded DNAs. This is what is going to happen in synthetic phase. Now when DNA replicates, many things are required. Enzymes are required, ATP is required, raw material will be required. So all that preparatory work will be done in G1. So what happens in G1 is, the cell prepares itself for S phase. So it is a preparatory phase. Then all that preparation will be used in S phase. In S phase, DNA will replicate, few more changes will take place. We will study it in detail also, but here I am just giving you a brief introduction of the cell cycle. So DNA replicates. DNA replication is a very complex process which requires 10 different types of enzymes, lots and lots of ATP molecules, raw material. The raw material is also of different types, various types of nucleotides and all those things. So all that preparation has to be done before the actual replication can take place. This is what is being done in G1. So once this preparation is done, the cell is ready to enter into S phase. In S phase, all those things will be used up, DNA will replicate, few more changes also take place here, and then the cell is going to go into G2 phase. What is G2? Gap 2. Gap word, whenever we use, it is for preparation. So in G1, cell prepared it for the next phase, that is S. In G2, the cell is going to prepare itself for the next phase, that is M phase. Now what happens in M phase? The cell divides. First the nucleus is going to divide, then the cytoplasm is going to divide. So everything what is required for this division, that has to be prepared. So here in G2, what is going to happen? The cell gets ready for M phase. What will be required? We will write down all those specific points later, but what is going to happen when we talk of preparation in M phase, or sorry, for M phase? In M, the nucleus is going to divide. Now nucleus doesn't divide like this. First, all the membranes should dissociate so that for the chromosomes to separate, enough space should be available. So all the membranes should dissociate. How will the membrane dissociate? There has to be an enzyme which is going to digest these membranes. These enzymes are going to break the membranes. We call them lytic enzymes. So enzymes are to be synthesized. Then later on, when the new cells are formed, we will have to make these membranes. All those organelles should reappear. So we would need synthesizing enzymes, synthetic enzymes. Then the chromosomes are to be pulled apart. The chromosomes will be moved to two poles. We will see how they move. But there are some fibers which are going to pull the chromosomes to opposite poles. These fibers are spindle fibers. You remember I gave you the label kinetochore in the diagram of chromosome? The spindle fiber attaches there, and this is going to pull the chromosome here, this is going to pull the chromosome here, and the chromosome is going to split. That means one chromosome will go towards one side, other chromosome will come towards other side, this will go in one cell, this will go in one cell. That means now every cell is going to have the same amount of genetic material, but for that the DNA has to replicate, which has happened in S phase. Okay, so what preparation? Spindle fiber. It is made up of tubulin protein. So these proteins are to be synthesized. ATP is required. You know, one chromosome, if it has to be moved from the midpoint to the pole, it requires about 30 ATPs. And we have 46 chromosomes. So 46 are going to move towards one pole, 46 are going to move towards the opposite pole. So 46 into 30 and 46 into 30, one cell dividing only once. So this is what is the amount of ATP that we are talking about. So all that has to be prepared, and everything will be done in G2. And with all this preparation, when the cell enters into M phase, everything is available. Complete division will take place. And after the cytoplasm divides, we will have two tiny cells. And these are the daughter cells. Now suppose a cell has to divide once. What is going to happen? This cell starts, G1, S, G2, M, and two daughter cells are formed. Now this cell is not going to divide again. What are these two daughter cells going to do? They have to grow to their normal size, and they should start performing a specific function for which these cells have been made. If a cell has to divide continuously like meristematic cell, then what will happen is this cell starts, gets into G1, S, G2, M, daughter cells are formed, they grow, again G1, S, G2, M, this is going to go on and on and on. But as I was talking about, if a cell is not going to divide further, let us take this one. These two daughter cells which are formed, these two, they are not going to divide now. So what happens? These two daughter cells, they enter into G1. They are tiny cells. Their cytoplasmic content is less. Their size is small. So nucleocytoplasmic ratio is not proper. So what happens is they grow. And then they come into a phase, and this phase is known as G0 phase. What is this G0 phase? G0 phase is a phase which is known as quiescent phase. Quiescence or quiescent means inactive, but here we are using the word inactive only in respect of division. So here the cells are not going to divide. They are inactive only for division, but they are specialized. The cells, they become highly specialized. Any cell of our body which is formed like this. So if a cell is to be formed, a special cell is to be formed, the cell is always going to arise from pre-existing cell. So that pre-existing cell gets into G1, gets into S, G2 and divides. Now these two daughter cells, they have to become specialized. So what happens is they come into G1, they grow to their normal size, and now they come into this phase, which is called G0 phase, G0 phase or quiescent phase, and they become specialized. So this change, when the cells come out of cell cycle means now they are not going to participate in the same G1, S, G2 and M phase. So they come into a stage where it is not a part of the cell cycle completely reoccurring. So it remains here, but that stage is called G0, and the cell is termed as matured, or it gets differentiated. The cell gets differentiated. It becomes a specialized cell. It can become parenchyma, it can become collenchyma, it can become liver cell in our case, it can become any kind of special cell. So now all those special cells which are there in any organism, they are formed in the same manner, and they are in G0, because now they are not going to divide. So when the cells are in G0, they do not divide. But In plants, there is parenchyma. Parenchyma cells, if required, can divide. So what will happen is, the cell has come into G0 phase, but now the plant needs those parenchyma cells to divide for some specific thing. So those cells will come back into the cycle. So what will happen is, those cells enter into again G1. The remaining changes will take place. That means now the cell is going to prepare for S phase, and the cell gets into S, G2, M, and if it has to divide and divide, it will continue in that cell cycle. We have not written down the exact points or the changes which take place during the cell cycle. I have given you a brief explanation of cell cycle. So before we write down all those things, let us take a quick recap of the important things. Okay, we started with the cell cycle, what exactly it is. It is the life cycle of a cell in which there are 3 stages. The cell is formed, it grows, and it divides. Then we talked about chromosome, because we have to remember that here when we are talking about division, the DNA has to replicate. Where is this DNA? It is in the chromosome. When we draw a cell, we do not label chromosome, we label chromatin fiber. So there is a question, what is chromatin fiber and where is this chromosome? Suddenly it comes from. So it is coming from chromatin fiber. How is it formed? It is the same structure, but after condensation, after dehydration, it becomes thick, and then we start calling it chromosome. We studied the detailed structure, the 3 important things. One was chromonemata, where the DNA is located. Then we talked about kinetochore, where the spindle fiber is going to get attached. Chromoplasm, from where the water is lost, and then only the chromosome becomes thick, rod-like, and visible to us. We talked about ploidy, because in the later part of cell cycle, when we talk of C value and which type of cell is formed, we will require this ploidy thing. Then phases, interphase, M phase. Interphase is completed in 3 parts, G1, S, and G2, and M phase is completed in 2 parts, karyokinesis and cytokinesis. We have to talk about all these stages in detail, which we will be doing in the following sessions, and this was the cell cycle. This is just a brief thing which I have given you. We have not written exactly what happens during each stage. This we will do in the next lecture. So this was just to give you an idea of what exactly cell cycle is, what are the phases, and when we meet next, we will be talking about each and every change which takes place in a cell cycle. And do you think that something can go wrong in this part? Suppose some raw material is not synthesized, sufficient ATP is not there, DNA is not properly divided or replicated. What is going to happen? Should there be some check here? So in the cell cycle, there are some checkpoints. That is why every cell in our body which is formed is a perfect cell. If something goes wrong, the cells will be mutated, and many times mutations result into cancer. So when division is taking place and everything is fine, that means there are checkpoints in place. So details of all the phases and checkpoints, that is what we will be taking in the next session. And don't forget to subscribe, and if you want to subscribe and take a plan, use or take that plan or purchase that plan with this code so that you are able to avail 10% discount. So I hope the basics of cell cycle are clear. I will see you soon in the next lecture. Till then, take care. Bye-bye.